Good afternoon. This is Dan Guerra from VerEvMed Studios from the Pacific Intermountain Northwest of the United States. Today we're going to cover an interesting topic related to a neuropathological disorder, Alzheimer's disease. Now, we've talked about Alzheimer's in the past. We talked about neurofibrillary tangles, tau protein associated with that. We've talked about the A-beta protein and plaques. We've talked about a lot of the neurochemistry and neurobiology of this particular disease. Today we're going to take a little bit of a different pathway. We're going to look at how epigenetics might play a role in the occurrence and, in fact, maybe in the diagnosis of prodromal Alzheimer's disease in humans. So let's get started, shall we? All right, this is VerEvMed, company we have. This is our website and the email you can contact us to be our client. That's me, Dr. Dan Guerra, and that is my particular email that you can contact me on any of these lectures that you see. If you want to talk about them, if you have any interest in me perhaps tackling a topic that you'd like me to cover, contact me and we can work something out, I'm sure. These are free to the public. Our company is a company that works with clients, and what we do is we bring the scientific literature to you, and we help interpret it, explain it, and help to see how the scientific literature may help you in many of the endeavors you're involved in. You as a pharmaceutical scientist, you as a physician, you as a surgeon, you as a veterinarian, you as a lawyer, or indeed anyone that is interested in how biomedicine impacts their life. If you want to know what kind of things are going on in the actual research, that is at the scientific level, that's what we do. Okay, so today's the 31st of March, 2018, kind of a special day, and you'll see why that is later. By the way, there's my Facebook page. You can go there and you can get all of these recordings free of charge on that Facebook page, plus bonus some really cool metaphysics. All right, so the topic today is, does epigenetics play a role in the etiology of prodromal Alzheimer's disease? So let's get started here. All right, so this is a paper that's in press, that's in a journal called Alzheimer's and Dementia. So let's take a look at what they say in the intro, shall we? Late onset Alzheimer's disease, I'm going to call AD, neurodegeneration, is prefaced in early preclinical non-symptomatic middle age with proto A-beta plaque and tau neurofibrillary tangle CNS pathology. AD remains largely idiopathic, so there are multiple lines of research to uncover the prodromal cellular biochemistry and cell biology of the disease, including the potential for epigenetics. Don't worry if you don't know what that means. I'm going to get into it in some detail momentarily. So an epigenomic-wide array was examined for AD. And what that means is normally you think about GWAS, which are genomic-wide arrays, looking at what genes may be mutated that are associated, let's say, with a given disease or syndrome. Same kind of idea here, only we're not looking at the genetics, we're looking at the epigenetics. So such screens are available and data is available online. And I'll explain to you again in more detail what all that might mean. But anyways, doing an EWAS, doing an epigenetic screening in AD patients, it was uncovered that there's a consensus neuropathic associated DNA methylation pattern, including a CPG hypermethylation in one particular locus, the ankyrin gene locus, ANK1, in fact. So when I say hypermethylation, what am I saying? That means a lot of CH3 groups added to that cytosine residue in those CPG dinucleotide islands found usually proximal to the open reading frame of the gene that's going to be expressed. That's one of the major epigenetic markers, molecular markers. Adding a methyl group to a cytosine residue in the DNA will alter the expression of that DNA. okay, adding that methyl group, especially if that methyl group is compounded by adding multiple series of methylation patterns into certain proximal promoter regions so that the gene is actually regulated to be expressed completely differently. That's why it's epigenetics, you see. So what about this anchorin locus, this anchors gene? Well, ANK1 anchors. What that protein does is gives you an idea, okay, when we're talking about what kind of genes get methylated that might be altered by that methylation. So it's nothing to do directly with the genetic sequence. It's not a mutation in the sequence. It's actually altering basically the expression of the gene in situ in real time when you add methyl groups to the cytosine and CPG islands in proximal regions of that gene that's being looked at. This particular one is ANK1. So what does ANK1 do? We need to know that if we want to have any idea about the molecularity of this methylation pattern. Well, ANK1 anchors, several integral membrane proteins to cohering cytoskeletal domains. So you have cytoskeletal proteins. They have certain structural domains. As part of a protein, it's actually what evolves are structural domains, not so much functional domains. Another story I'll get into sometime when I talk about gene and protein evolution. At any rate, it appears that ANK1, what it does, it anchors other proteins to these cytoskeletal domains. So you have basically a grid of proteins in the cell. So I looked up some of the functions of ANK1 just so you get an idea what we're talking about. So here are some of the things. It's involved in cytoskeletal organization, like as I just said. ER to Golgi vesicle mediated transport, kind of important, right? Moving membrane proteins and membrane lipids. Exocytosis, that is sending things out of the cell. Anchorin 1's involved. Maintenance of epithelial cell apical basal polarity, also very important. I think some of you might see how that could be associated with cancer. Positive regulation of organelle organization. Wow, very important. Mitochondria, peroxisomes, ER, Golgi, etc. Protein localization to the plasma membrane. You can gather that from all the previous bullets there. And because of all of that, signal transduction cascades. So anchorin, you might think, it's kind of boring. It's an anchoring protein. Look at all the things it really is involved in. But you see, that's going to be upstream from anchorin's expression, right? So, downstream. All these events are going to be downstream from anchorin's expression. So when we're talking about methylation of anchorin, we're saying that will be upstream from all of these potential biological events. You see? Of course you do. All right. Now, in situ, chromosome 7, human chromosome 7, linked Homeobox A, that's a different gene cluster now, a different gene locus, differentially methylated positions, DMPs, have been linked to prodromal AD. Now, we just talked about anchorin. Okay, that was one example. What kind of genes get methylated? What kind of genes have epigenetic signatures? Well, we said ANK1. Now I'm introducing a new topic, and that's actually the topic of today's paper, the HOXA gene. HOXA stands for Homeobox A. Don't worry so much about that. These genes were named in Drosophila, the fruit fly. But Homeobox genes are basically transcription factors that control development, okay? Animal development, for example. Embryonic development in particular. So anyways, the Homeobox A gene on chromosome 7 seems to have differential methylation positions. Those are called DMPs. Acronyms are loaded with genetic discussions. I told you the geneticists love acronyms. There is an extensive HOXA cluster methylome. That means a specific group of CPG islands which are known to be methylated. And that is linked, that methylomic pattern is linked to AD, associated neuropathology. All right. HOXA was described in the murine model, that's mouse, as an 11-gene cluster called the Homeobox region A, which upon deletion analysis, so when you want to know what a gene does, you carry out partial deletions, and you say, what did we lose when we did the deletions? Okay, there's other ways of doing that, but that's what deletion analysis can do. Anyways, when you do that with the whole HOXA gene cluster, and look, there's 11 genes in there, so it's a lot of things, a lot of work there. It appeared responsible for patterning the primary and secondary axes of the developing embryo. And you get all that information from this free website, Gene Cards. Okay, so I just looked up this gene in Gene Cards, and I'm telling you what HOXA does. I already told you what AG did, right? So let's get more into detail about HOXA. There's a rare genetic disease in humans with mutations within the HOXA cluster called the Curarino syndrome. Curarino syndrome, characterized by a triad of anal-rectal malformations called ARMS, usually anal stenoses, pre-sacral mass, commonly anterior sacral meningocele or ASMs, and or teratoma, and the sacral anomalies, that is, total or partial agenesis. And agenesis means a failure of developmental processes leading to organ terminal differentiation. See, A before it means negate. Genesis means the development or the generation of. So an agenesis in developmental biology means the failure of developmental processes leading to organ terminal differentiation. And that's what happens when you corrupt the HOXA cluster. Okay, that's what we're telling you here. And it's found in this particular syndrome, which is an inborn error of metabolism called the Curarino syndrome. Okay, I can't roll my R's, sorry, even though I'm Italian. Okay, now the acute, that's one disease that's been linked to corruptions in HOXA cluster. Remember, it's a transcription factor, shouldn't be a surprise, you get some global effects. Second disorder that's been linked to HOXA, acute T-cell lymphoblastic leukemia. Wow, we've heard of that before. I've talked about it in VerEvMed lectures. It's also associated with HOXA mutations, where it's been observed that the HOXA expression controls and can predict response to the BCL2 inhibition in acute myeloid leukemia. Okay, the BCL2 is normally the gene that we think about when we think about lymphoblastic leukemia, and indeed, it looks like HOXA is involved in the regulation of the BCL2. Okay, that was from a leukemia paper 2017, which I looked up. Next thing to know about HOXA. In general, HOXA are a family of transcription factors working upstream of several myeloid biogenesis factors, including those associated with hematopoiesis, and when inappropriately expressed, for example, overexpressed, are the genetic determinants for self-renewal of leukemic stem cells. Hence, you can see the linkage then to lymphoblastic leukemia. Now, I got that information from an Oncotarget paper published in 2013, and also from a Hematologia paper published in 2016. So these are the papers I've read to prepare for this talk, and that's where I got that information from. So you get the idea now what HOXA is about. It's all over the literature, not all over the literature in AD. That's today's paper. All right, so take a look at the embryo itself, the developing embryo. This is a 33-day-old human embryo. And you can see here the neural crest streams emanating from the rhombomers 1 through 8, numbered here. Okay. And these are influenced, these rhombomere regions are influenced by the Homeobox gene expression patterns. So look here, there's HOXA2 is involved in the development of this region. HOXB2 overlapping involved in this region, this R2, R4 region here. HOXA3, B3, and D3 in this region down here, right? The hyoid stream. So this is the frontal nasal sheet in early embryogenesis. This is the trigeminal stream. Those of you that have taken development biology know what I'm talking about here. This is this is the hyoid stream, and there's the postotic stream down here. Okay. All right. So you see where the Homeobox genes are important as transcription factors regulating the development of the human embryo at day 33. Okay. And these, FB stands for forebrain, E is eye, midbrain, etc. That's where those organs will develop once the embryo develops. Okay, this is before those organs fully develop. So you see why if you have a corruption in HOXA, why you get that agenesis, right? Because you don't get the full development of the organ. You don't get full development. Normally these embryos would die, in utero. All right. So that's about the logistics. I want to talk now about epigenetics, okay? So we have an idea that we're going to be looking at methylation patterns. we have an idea where we're looking in the embryo, the developing embryo. Now, we're also going to be looking at fully developed people and fully developed brains, right? This is Alzheimer's disease. But now I want to explain more detail of what I mean by epigenetics. So before we actually do the frank discussion of the molecular aspects of it, let's talk logistically, just in terms of the biology, where we're at, okay, for those of you that don't have a strong background in this. Okay, very simple stuff. Every cell has an identical copy of nuclear DNA, but the expression of the genes is controlled by the activity of a region, usually proximal, that means before, called the promoter region. Okay. Now, it's much more complicated than that, so these are very, very, very basic tenets in molecular biology. Now, the promoters are controlled themselves. Now, remember that DNA by a vast array of transacting factors. Those are usually proteins. Sometimes it's also nucleic acid and protein, but usually proteins. Transcription factors are usually proteins, which bind DNA and covalent modification. Okay, so what happens here is that you get a binding of these promoter regions, okay, by protein, and you also can get covalent modification of the promoter region. Okay, there's two things going on here. Transcription factor can bind, but that's not a covalent bond usually. If it is, it's more like a hydrogen bond or hydrophobic interaction. But you can actually get covalent modification like what? Most common covalent modification is the methylation, CH3 addition, of a cytosine in cytosine P guanosine islands, CpG islands, proximal to a gene which will be expressed in the promoter region. Now, it can be in enhancer regions, which can be differentially separated within splice sites within a large exome intron full gene cluster. But just for purposes of this talk, very simply in the promoter region. So if you add that methyl group, that promoter region, in general, it makes it so that that gene is retarded in its expression. There's less expression. Because why? Because when the RNA polymerase comes along and is looking for a promoter region to start transcription site, it's going to find a methyl group on those cytosines. And if there's a lot of them, basically the RNA polymerase can't bind very effectively, very potently. And you don't get the transcription, or not much of it. That's the idea of this, okay? So the methylation of the promoter DNA shuts transcription off. And this called the silencing, as it was used to be called in the literature, can be transferred mitotically and indeed meiotically. So germline transmission and also just regular cell division. So there are methylation patterns which can stick, which can stay. You know, that's because of how DNA replicates. That's not always the case. So you can add a methyl group, right, by a methyltransferase enzyme to a specific region of a CpG island, a specific promoter, based on sequence, topology, and fluid chemistry of the DNA and the protein that's adding the methyl group, and the availability of the methylating agent, which is usually acetylcysteine with thionine, by the way. But even saying all of that, it doesn't necessarily mean you get that methylation. Okay, so the methylation itself is specific. It's not just some stochastic random thing. In fact, it appears that the epigenetic modification of those cytosines relates to basically the DNA living through time, right? So the DNA has a methylation pattern in a unique sense, in a unique situation per cell, per tissue, per organ, per organism. So you and your twin, if you had a twin brother, may have exactly almost identical genome, but your epigenome can be quite different. Because let's say your brother grew up in, the Sudan, and you grew up in New York City. Just the environment can alter the methylation pattern of those promoter regions in ways that we don't fully understand, why some genes get methylated and some don't. We think it has something to do with over or under expression in times of important signal transducing occurrences in that genome where rapid changes in gene expression are required for, say, survival, or for something down the road that needs to be meted out. Let's say the arrival of some new system, like a developmental system. But that methyl group can be removed too, because there are demethylases as well. And that also is regulated, right, by the environment and also by the in situ biochemical molecular machinery within the nucleus. So we're just talking nuclear DNA. And I remember there's other DNA in the cell, like mitochondrial, and plants also have chloroplast DNA. So we're only talking here about nuclear, where most of this work is done. Now, besides this phenomenon, gene expression is also controlled via chromatin remodeling and ubiquitin, which is a totally different set of scenarios, which I'm not going to get into right now. But we have talked about chromatin remodeling, and we've touched you on ubiquitin several times in these lectures. So I don't feel guilty about not going through them again right now, because you can go back and look at them, or you can get any other source, such as a textbook or some other, maybe even video and a way of explaining it. So I'm not going to do that here. Now, all modifications to genes other than changes in the DNA sequence itself is epigenetics. Epigenetic modifications include the addition of the methyl groups, CH3, to the DNA backbone. Adding those groups changes the appearance and structure, so it's a physical chemical property change in the DNA. So that makes sense, because you change the structure, you can change the what? What am I going to say? Function. Remember, structure-function relationships, really big in biology. This changes how genes interact with one another and with other molecules in the cell nucleus. So there's epistatic effects. There's gene-gene interactions because you've had the methylome of one over the other. Because sometimes you have multiple copies of genes. Some copies of the genes are silenced by methylation, and some are not silenced. This is what happens when you have identical copies of genes coming from the mother and father. So you have a control over which of those parental gene clusters are expressed in the developing embryo. And that helps coordinate the developmental pattern, and also later on in the life, in the entire life of the organism, right? Even all the way through adulthood. All right. So let's get more frank and serious about epigenomic considerations. Let's consider epigenomics, shall we? The epigenome is that layer of genetic information supervening onto the DNA helix, and they may be particularly prone to random changes than in its DNA sequence counterpart. Now, I use the word random, or you could say stochastic if you want to sound clever, because it may be random in the sense that it could be provocative associated with environmental changes, such as stresses, such as, let's say, regular chronic intoxication with ethanol. Yeah, okay. Now that's random for the cell. The cell or the system doesn't know that ethanol is going to be taken on board. Or any other possible thing, maybe just normal stress, maybe just overthinking a project or feeling melancholy. Those kinds of things could be called random in the sense of the normal functioning of gene expression, not random in the mechanism itself. So I want to make that clear. All right? Random in the sense that, again, you got the two twins, the one in Sudan and the one in New York City. Whatever events happened to the guy in Sudan, right, living along the Blue Nile during a raid from some, terrible warlord, that's going to be a different stress than, let's say, the hipster living in New York City that's going out to nightclubs every night, right? So that's what I mean by random, not the kind of thing you can predict. As in the case for the DNA sequence of the genome, the maintenance of the epigenome is a challenge for the mechanism of repair, recombination, and replication, as well as for transcription and then ultimate synthesis of the protein and whatever's going on in metabolic systems. So when I say it's a challenge, it's because this epigenome lays another layer of complexity on it. Okay, a very important one. So now let's get into the background. Genetic and epigenetic mechanisms shape metabolic activity. This is what you find in the literature. That's what I mean by background. Now I'm telling you, I'm after I've vetted the literature, because I've worked in epigenomics now for, going on about 15 years. And I saw it when it first came on board, and now I see it maturing quite a bit. So this is a summary of that, and I call it a background to understanding why we're so interested in epigenetics and why this paper is significant when it relates to Alzheimer's disease. Genetic and epigenetic mechanisms shape metabolic activity and can respond negatively to produce a pathophysiological state. Okay, I think we can understand that if the methylomic program alters gene expression in such a way that you don't get defense genes being made, let's say pro-inflammatory cytokines, you can see where that causes a problem. Or if you get an overexpression of pro-inflammatory cytokines, you get autoimmune disease. See? So while the genome establishes the template for developmental and metabolic patterns. An adaptational phenomenon helps to produce the final phenotype, and that's the epigenetics. That later epigenetic mechanism has become a key subject of developmental and cell biology, gene expression, and indeed disease. The biochemistry of epigenetics involves several covalent modifications, as we've been saying, nuclear chromatin, particular DNA, and the histones. I talked about the histone code before. So those cohering basic proteins that wrap around and make nucleosomes within the eukaryotic nucleus, those proteins called histones, okay, there's 8 of them. Though that octet of proteins can also be acetylated and methylated, giving you another layer of epigenetic reprogramming of gene expression. Yeah. As well as post-transcriptional RNA-based gene silencing. So what we're saying there is you also have interfering RNA. Right? So it's an interfering RNA is also loosely called epigenetics. It's a whole other story that I'm not going to get into this afternoon. These modifications can be reversibly administered by interactions with the genome caused by the resulting in poor nutrition and lifestyle, or such things as like chronic ingestion of drugs, or the drug alcohol, or other kinds of noxious substances like that. And there is a spatiotemporal pathological metastate generated. In other words, the methylation can occur in a certain location during a certain temporal sequence, and then it can be removed. And unless we're there viewing that on-off methylation, we never knew it happened. Yet we can see big shifts in what? Gene expression. So it's not always that easy to detect what's going on. So the players can be very subtle. In fact, the discovery of epigenetics took this long because we didn't have the tools to investigate. Now we do. Okay. So again, let's go real quickly here because it's just more background. Among the modifications are specifically on the carbon-5 atom with cytosine residues found in certain canonical CpG islands. We've already gone through this. There's also the processing of double-stranded RNA and the generation of small interfering RNA involving gene silencing. Mechanisms include the methyltransferases. I told you there are enzymes that do that. Acetyltransferases. We've already talked about deacetylases. Remember the sirtuins. Talked a lot about that. Kinases, adding phosphate, phosphatases, removing it. Demethylases, deacetylases, ether ubiquitin ligases, and etc. The substrates for all of those reactions are either chromatin, or in the case of the RNase activities, double-stranded messenger RNA. Acetylmethionine, or also known as ADOMET, is the nuclear methylation agent, and acetyl-CoA is the acetylation agent. Okay? Nuclear-associated post-translational modifications, such as acetylation of histone carboxy termini, clearly alter chromatin structure and function. Basic take-home about acetylation, if you want to just know something, boom, like a heuristic. When you acetylate histones, you open up the DNA so that it could be replicated, recombined, repaired, and transcribed. When you remove the acetate, you condense that DNA, causing the formation of a structure called heterochromatin, and then you get less overall replication, recombination, repair, and indeed expression and transcription. That's just a general heuristic. The major effect is a pronounced change in the physical-chemical accessibility of DNA binding proteins to unwind the double helix and essentially potentiate transcription of RNA. This is a big deal, is what we're saying. Finally, let's introduce now this paper. The human phenotype is influenced directly or in combination with our genes and the environment. Again, environment meaning the cellular environment, right? We're not talking about whether or not it's sunny outside, although things like solar radiation can impact the epigenome. There is, not necessarily though, that's why we call it stochastic. There is a genetic predisposition to many diseases, but how are environmental stimuli involved mechanistically with the phenotype? This is now getting to the core of how some people get sick from some external stimuli and some don't. Epigenetics, you see, is the study of changes to gene expression independently of genotype. I would put that in quotation marks, independently, because genotype does play a role. Otherwise, you wouldn't be talking about CpG islands, localized in promoter regions, right? That's not independent of genotype. But it is independent of the gene itself, because usually these are in what? Intervening regions, not in exons. Right? That's not always the case, of course. In certain instances, these are transgenerationally inherited. I Told you that. You can inherit your epigenome. You can inherit your grandfather's epigenome. You can inherit your grandmother's epigenome. And that means that there was a methylation pattern that could have been set in motion 60, 80 years ago. Yeah, because of some environmental stimuli. Let's say drought on the Transvaal in Africa, or let's say starvation, like in post-World War II, the Netherlands. Yeah, that kind of thing. All right. So this then helps specify tissue-specific and temporal gene expression. It helps us. It helps specify when those genes are expressed in time and place. It also helps us get a handle on how this might be regulated. All right. So aberrant epigenetic profiles are associated with numerous diseases. Environmental stimuli may induce those modifications and in turn modulate gene expression. Furthermore, the microRNAs impact protein abundance through post-transcriptional regulation. Again, bracketing that off for now. Physical exercise is an environmental stimulus that, of course, impacts health and well-being. The more exercise you get, usually the better it is. And that actually is a potential trigger for epigenomic remodeling. I find a lot of papers in that subject recently. So the health and fitness benefits conferred by regular aerobic and resistance exercise training include alleviating risk of developing and reducing severity of cardiovascular, metabolic, pulmonary, hepatic, renal, and immune disease. Obesity and cancer are other obvious pathologies impacted both phenotypically and, here we go, epigenetically by exercise. This is all found in the literature, okay? Exercise does impact the signatures of the epigenome, including DNA methylation, which we're talking about here, histone acetylation, etc. So you see it. All right, let's take a look at this now. Let's say you have normal genetics, which equals inheritance, which equals maybe a determination of, like, say, hair color, eye color. Then you get environmental influences, which can alter that gene expression, and you get a phenotype. This is your standard genetic model. With epigenetics, though, you get some non-inherited epigenetic determinants, which can play back into environmental influences. You get genomic imprinting, which is another form actually of epigenetics, the imprinting from generations back through meiotic selection, right? You inherit all of that, which determines, and you get a new phenotype. So what we're saying is that the inherited epigenetic determinations are supervening on the genetic genomic framework for the given phenotype. So far more complicated. Look at all these arrows. Some of them are going in both directions. And we don't really know all the intermediates in these pathways. We're just getting started with this, even though we've been doing it for 15 years. Now, I hesitate putting this in the talk, but because you can watch this talk and you can slow it down and stop it, I wanted to just make sure that you knew all these terms: chromatin, DNA methylation, etc., the epigenome. I'm not going to go through them, but this is for your reference. If you want to make sure you go back and you listen to someone like me talk, you're saying, well, what exactly does he mean by histone acetylation? Here is a little heuristic bullet definition for those things. All right, let's get back to the paper. Finally. Alzheimer's and dementia. No problem about that here. For this research paper, the cohort consisted of brain tissue from 147 individuals obtained from the Mount Sinai Alzheimer's disease and schizophrenia brain bank. So these are from brains from what? From dead people, okay? From the 147 donors, 2 cortical regions, the prefrontal cortex and the superior temporal gyrus, the STG, were used for the purposes of the studies. This is like the materials for the research. Okay, I'm just giving you like the materials and methods section here. Very truncated, of course. Samples were randomized with respect to age and gender, and the analysis covered an average of 17 CPG sites per gene region, distributed across promoters, untranslated regions at the 5' end, the first exon, the gene itself, the open reading frame, they call it the gene body here, and the 3' untranslated region. And that covered about 96% of all the CPG islands, with additional coverage on island shores that is adjacent to the islands, and even the flanking regions to those shores. So they really tried to throw out a wide net here. They had 147 individuals' brains, so they had a lot of tissue to work with. So they really wanted, when they got this tissue, which is, of course, a great gift to be able to get this, who donated this, these people who donated this for science, you want to be very careful to use that very precious tissue. with great care, and you want to treat it with that kind of attitude. But also you want to get as much information as you can, because this is the kind of thing that just grows on trees. You're very, very lucky to be able to get this tissue. So you want to be able to get as much research out of that tissue as you can. So they did a very deep measurement of the epigenome across those regions. That's what they're telling you here. So once again, I just want you to get an idea here. Within the nucleus, you have chromatin, and chromatin can be remodeled by methylation and acetylation, wrapped up with histones that are called the nucleosome. Okay, you can have an increased abundance or a decreased abundance of this whole system depending upon the methylation pattern. DNA methylation itself can be up or down, and it can alter transcription, for example. And because if you alter transcription, you'll alter translation. Alter translation, you alter the amount of protein. The protein itself can modify and go back all these other regions if that protein, for example, is a transcription factor, right? Such as the Hox genes. Yeah. So it just gives you like a pictorial of what we've already said. Again, probably not necessary. Now, how do you go about looking for the methylome? Well, I can tell you exactly how, because I've done this in the laboratory several times. It's called the bisulfite conversion assay. And basically what it is that if you have a methyl group on a cytosine, you cannot modify that group with bisulfite. You can't change the structure of it by using bisulfite. Because of that, after replication, the methylated genome will be the same sequence before and after you use PCR to carry out the replication, or otherwise known as PCR amplification. So you have genomic DNA, you convert that DNA, you take that DNA and you chemically modify it with bisulfite, so that the bisulfite's only going to be able to bind to those cytosine residues that are not methylated. You PCR amplify by using primers to that region. Then you get what's called a tagmentation. You sequence that library, and you read the quality trimming and the mapping, and you calculate the percent methylation directly from this sequence, okay? So this little dot here is 5-methylcytosine. That's what not modified. It couldn't be modified by bisulfite. There's your target region. That's what they call tagmentations. And there's the sequencing adapters and indices to be able to look at specific regions of that DNA. So that's basically the schematic of the bisulfite method. And again, it's looking at genomic DNA. You treat it with bisulfite, you convert it, and you modify the unmethylated, right? The unmethylated with the bisulfite. Methylated doesn't get modified. So how does that work out? Well, basically, you set up a read depth for either 100% or 0% standard. So if you got the CPG islands, see, you'll put the P in here, you don't need it, right? There's a CPG island, there's another one, there's another cluster there. And if there's no methylation, what happens is all of those C's get converted to T's, right? So round of replication, the PCR polymerase reaction, you get all T's here. This is a 0% standard, no methylation. The 100%, all of those cytosines on carbon 5 are methylated. Okay, that they've been manufactured that way to generate this 0%, 100% standard. This is a standard for your analyte that's coming on board. Then all of those are going to stay as cytosines, right? Why is that? Because they're methylated, right? So they can't. Whenever you go through whenever you replicate, you're going to the next round of replication. Those will be turned into thymine, but not if they're methylated, okay? Because of the bisulfite. You see. Very interesting. So that's what you got, a 100% standard, all methylated, and then you got a 0% standard, no methylated. So it's examples of sequencing outputs for methylation controls in silico converted reference sequences. So this can all be determined in silico with your computer. You have a sequence. You say, if all of those CPG islands were methylated, what's it going to look like? Boom. If none of them are, what's it going to look like? And this are your frame, right? This is your bookend for what your data is going to fall within. Heck yeah. All right, now here's some real data. So the rhodopsin protein, relative messenger RNA expression from mouse, this is a mouse system here, cerebellar and retinal tissue, okay? Now, relative expression of rhodopsin in the retina, what? A boatload of it. It's rhodopsin. That's what? That's the pigment protein complex in the retina. How about the cerebellum? Boy, you can't find any. Well, why is that? Check this out. The 5-methylcytosine content of rhodopsin, the cerebellum, is really high, whereas in the retina, nothing. So the methylation of that gene regulates the expression of it in space, that is in the retina versus the cerebellum, and probably also in time because of development. Okay. Now down here, we're looking at CPG sites relative to the transcription start site, and we're looking at either the retina down here. You don't have much methylation, but there is a little bit of scatter in how much you get down there. But look it up here. Even within that particular gene, the rhodopsin gene, relative to where that CPG island is, you get a different pattern of methylation. So it's not so simple. You say, well, I got these CPG islands and they're all going to either be methylated or not methylated. I got to figure it out. I go to get my DNA. I do the bisulfite treatment. I see that my DNA has 33% non-methylated versus methylated. And I say, well, that means 33% of that particular PCR abundant fragment that I just amplified is methylated. Not good enough, Charlie. You know why it's not good enough? Because there are multiple CPG islands, right? Far more complicated. So what you need to do, and I know why this is complicated because I did the work and I had a lot of help doing this, but you had to go in and read sequences and you had to look at where CPG clusters were, and part of it was with the computer algorithm that helped, but the other is just by using your mind, looking at where those CPG islands are, and then saying, how can we predict which of those islands may or may not be methylated? You can't predict it unless you have a database, and we do have databases like this one. Right? So we have an idea, because of this data, which of the CPG islands will be methylated and which won't, even compared to, say, the cerebellum versus the retina. Very cool, right? So this data is becoming more and more available, but it's still fragmentary compared to like just genomic analysis. This is epigenomic analysis you're looking at here. But you see how this works. Another layer of complexity. Every time you add a layer of complexity, you're not going arithmetically in the complexity, you're going logarithmically, right? Heck yeah, especially when you think about multiple sides. But it's doable. It's doable. Now, I know this is a really busy slide, but I love it. All right, it's just showing you the bisulfite amplicon sequencing. This is how it's done. You get the bisulfite conversion. Now, wherever you have the methylation pattern, see on these cytosine islands versus when you don't have it, right? You're going to convert all these cytosines, right, to thymines, right, because these were not methylated, but because you treated with bisulfite, right, all of these are going to stay as cytosines, right? You're going to do a dual index library. You're going to have all of these fragments that are methylated and all those particular very critically associated CPG islands in a particular promoter, a particular gene you're looking at. And likewise, CPG islands that are not methylated, you're going to microsequence the whole bit. You're going to get two different reads. You're going to get in silico bisulfite converted references now. Where do you get methylation? Where do you get the highest amount? See, these are statistical bars here. So where you're going to get the most of that methylation, and according to this data set, is that CPG island, and much less on this region and that region, you see? So this is tractable. Science is tractable. Likewise, it's saying the same thing on this side over here. So there's your 0 to 5% methylcytosine reference, and there's your basically 100%. Okay, if they stay as cytosines, these are all thymines. See how that works? All right. Now, of 78 differentially methylated positions, they call them DMPs, 9, and now we're back to the data, 9 were located in the HOXA gene cluster on chromosome 7, is where it's found, most notably in the vicinity of HOXA3. Now think back, HOXA3 is really important in that early development of the embryo. Now, wait a minute. We're talking Alzheimer's disease. Alzheimer's disease back at the embryo? I'm not saying that. I'm not saying it's not either. Okay? But I'm not saying that. What I am saying is that the HOX gene is involved in development of the brain and of the neural crest during development in the human embryo. But it's also involved in neural architecture in the adult. Okay? That's it, right? That's the point, okay? The gene cluster didn't go away because you went through the embryonic development. That's what I'm talking about here. So, okay, most notably in any of the HOXA3, this is the data from those 147 tissues we got from those specimens that were donated to science. Within one HOXA differentially methylated position reaching statistical significance. Only one. So of all the ones they looked at, remember all those different clusters they looked at, they only found one that looked like it really was statistically significant. So they identified a total of 6 methylated regions within the HOXA gene, with the most significant in a certain region, 364 base pair within intron 1 of HOXA3. So they really want to key in on that one region. That's where they found the most difference between the methylation pattern of all that HOXA gene cluster and whether or not it was methylated. So cortical neuropathology associated hypermethylation, cortical neuropathology, that means they had the brains from people who had Alzheimer's disease. Now they could have done this with what? With the schizophrenics too. In fact, somebody probably is. Remember, these brains are in a clinic for AD and schizophrenia. I'm not saying those diseases overlap. I'm just saying you've got brains from humans who had been diagnosed with schizophrenia that you can do the same analysis for. Yes, see. Okay. Cortical neuropathology associated hypermethylation of the HOXA3 gene at BRAC stage 3. Now you got to talk about BRAC stage, is observed in independent studies. Now, when you look at the different staging of Alzheimer's disease, there is a BRAC index. So the higher the BRAC stage, the more involved is the disease. Okay. So it's stage 3, which is what they're keying in on here, actually. Remember, this is a spatial temporal methylation pattern. Told you it wasn't going to be non-complex. Stage 3, what are we looking at here? This is the brain here, okay? Stage 3, lesions in the hippocampal formation worsen. Entorhinal becomes strongly involved. Lesions extend through the transentorhinal region into the adjoining higher order sensory associated areas of the temporal neocortex. Okay, so this region now is becoming more and more damaged in this Alzheimer's patient at BRAC stage 3. That's where we're getting highest level of this methylation pattern in that particular 364 base pair region in the HOXA3 gene. Okay? So this data is from active neuropathology. This is just looking at stereochemistry of brains. I put this in here to show you what this paper was talking about. They didn't do these cross sections. See, these aren't whatever these are, sagittal cuts or whatever. That's not them doing this work. I'm showing you so you get an idea, so you get a visual of where in the brain we're talking. So the entire basal surface of the temporal lobe reveals the severe involvement of the entorhinal complex. Severe meaning you get all kinds of damage. That's what the dark staining is. Anterior portions of the parahippocampal gyrus at stage 3 shows the tendency of the pathology to extend from there to the adjacent neocortex. That includes the occipitotemporal gyrus, where the arrows are shown here, and the lingual gyrus posteriorly, also shown down here. Okay, so you're starting to extend now. That's severe BRAC stage 3 AD neurocellular tissue pathology. Okay, wanted to show you. They mentioned it in the paper. I wanted to see what it looked like, and I wanted to show it to you. So I pulled this paper from Active Neuropath. There you go. All right, here's some more data from the paper. I don't like the way they presented this, but I put it here because I think it does show you what this data looks like. The data is not that easy to interpret. You have to know what you're looking for. Now, what they did determine, I'm just telling you this, okay? No probes meaning the probes on specific, using a PCR fragment to be able to nest into a gene, move into the gene and ask where that probe will pick up a CpG island after the bisulfite treatment. That kind of probe. No probes reach significant threshold in the cerebellum or in the pre-mortem whole blood. Okay, so they had blood from these people before they died. Obviously, it's got to be pre-mortem, right? Otherwise, it would just be coagulated blood. So they didn't find any of this stuff in the blood. They didn't find any of this in the cerebellum. So that indicates that the methylation pattern observed may be specific to only those particular cortical regions. Which ones are, again, the prefrontal cortex and the superior temporal gyrus. And that's what they're showing you here, okay? So these are different methods to analyze the data, Okay, different types of algorithmic screening techniques to look at how much. This is a lot of methylation, okay? You can see how it spreads out in this general region down here, but it's much less of it, but it's more spread out. And here is a single silo effect of methylation at a specific site, probably around that 364. This is counting actually at the genomic location, so you're not going to get that numbering system. So it's just genomic location. And these little diamonds here are showing you where you get the abundance of that methylation, okay? Prefrontal cortex and the superior temporal gyrus. That's what this data is showing you. So we can summarize the data, okay? I didn't show you the other figures, but they pretty much look the same way. Locking in a 364 base per position and HOXA3 in those two subregions of nuclei of the brain of these people who had died and were Alzheimer's disease tissues. The research data in this paper identified an extended region of elevated DNA methylation in the HOXA gene cluster, which I've already said. It's associated with AD neuropathology, I showed you that, with consistent effects seen across 3 independent post-mortem brain sample cohorts. These are entire cohorts that they looked at. Hypermethylation found within some 208 differentially methylated regions. Okay, so they looked at a lot more than 208, but they found at least 208 that had differential methylation, which means a change in the direction of expression of that transcription factor known as HOXA3. Differential DNA methylation in the HOXA gene cluster was noted in some Down syndrome. Now check this out. Individuals who have a tendency to eventually develop AD. So some Down syndrome people get AD later in life. And that Down syndrome, of course, is due to an additional copy of the APP gene because of the trisomy on chromosome 21. So you see, now we're starting to back up, get to the 20,000-foot level, or maybe because we're in the Rockies, the 14,000-foot level. Of course, we're not in Colorado right now, but thinking about the 14ers, missing them. At least looking at them anyways. What am I saying there? I'm saying that in a totally different neuropathology system, Down syndrome, looking at trisomy, looking at the multiplication of a particular gene, guess what? There's a DNA methylation pattern in that HOXA3 gene cluster too. So that means some of those pathologies could potentially overlap because the Down syndrome people tend to have a higher frequency of AD than non-Down syndrome people. So that's interesting. There's a correlation there way up at that elevation of the HOX gene in terms of methylation pattern. HOX genes may provide neuroprotection, so methylation at those loci may have something to do with the link to AD. So in other words, when you methylate those genes and you lower expression of them, you may lose the neuroprotection. That's the argument. Because certainly that's what you see in the tissue. Right? That doesn't mean that other things aren't going on. You bet they are. But what they're trying to say is, if we're looking for epigenomic patterns, we're not finding it in this, at least in this study, in the tau protein. We're not finding it in the A beta protein, okay, or the precursor protein. We're finding it. They found it in this really interesting developmental gene, homeobox gene system called the HOX genes, particularly A3, particularly the prefrontal cortex and the STG. Since HOX transcription factors are linked to anchor in gene expression, which I told you way back at the beginning, and methylation at both loci are associated with Alzheimer's disease, possible there's a hierarchical methylome signature surfacing in AD epigenomics. So you got methylation pattern in the HOX, and HOX control ANK expression. Both of them are methylated. Maybe there is an overlap there. Methylation along that corridor may be prodromal, way before you see disease symptoms, linked to a person that might well develop Alzheimer's disease, later onset Alzheimer's. There appears to be a neuropathology-associated hypermethylation of the HOX genes encoding transcription, which are transcription factors involved in neural development, as we said, and the link to AD is becoming more, as I like to say, transparent. Here's the dialectic. Genetic association with AD neuropathology is elusive, while biochemical endophenotypes have been extensively deciphered and demonstrated. So we don't really know what's causing it. Let me translate this, okay? But we do know after effects, such as the fibrillary tangles and the plaque. We know a lot about the biochemistry about that. Oh boy, do we. But we're still trying to link together how do you get there. That's what this point is. So premise one. Premise two, epigenetic encoding in neurological disease has been widely speculated, but only sparingly demonstrated. But now, okay, here's the conclusion, okay? Synthesis, sort of antithesis, because this is like epigenetics and genetics. The two aren't necessarily, they aren't the same thing. So you got genetics association. You maybe have epigenomics association with AD, although it's more speculative. Now I'm saying the synthesis of those two premises. What is emerging in AD research is a temporal synthesis of genomic by environmental inducers, whatever those inducers are in the environment, that are potentially linked through epigenetic modification, precipitating otherwise occult modifications, that is, you don't normally see them, of juvenile developmental paradigms, such as that whole Hox gene we saw there in the early embryo, leading to age-related neurological disease. Okay, so that's my synthesis of all the papers I read for this talk, and particularly trying to put this paper in its context. All right, now before we leave, I cannot let this go. Today is March 31st, and I have to recognize two very important people that were born on this day. One of my major muses, both in philosophy and in science, is Rene Descartes, the French, basically one of the probably the 20th smartest people that have ever published papers, at least, right? A polymath for sure, emphasis on math, right? Today, we celebrate Rene's 422nd birthday. He was born on this day, March 31st, in 1596 in France. Remember, he came up with I think, therefore I am, cogito, the cogito or cogito ergo sum. Let's talk a little bit about him, not just say it's his birthday. I want to make sure that you know a little bit about Rene. Why not? Descartes was a rationalist conceptualist in terms of philosophy, which means the individual possesses intuitive knowledge of basic principles. That's what it means to be a rationalist, and a conceptualist as opposed to, let's say, a nominalist or a universalist. He believed a deductive system for self-evident truths, as in math, because he was a mathematician by training. So axioms, as we know, lead to deductive proofs, and that's how the inquiry proceeds, from deductive systems to getting evident truths, self-evident to you, the self. He says that logical certainty is obtainable as in geometrical proofs. So again, coming from a mathematical model, mathematical training. He was a dualist, which he suggested was mind or soul is immaterial, while the body is obviously physical, and it's basically material, right? So this is non-material. So he was one of the first dualists. In fact, as we will find out later on here, he's basically the father of modern metaphysics, Western metaphysics. Okay, now, besides this, Descartes was a libertarian. Libertarian doesn't mean a political affiliation. Forget about that. That means you believe in free will of choice, which means you're a non-determinist, okay? Free will. After all, he was a Roman Catholic, and us Catholics believe in free will. Descartes is also from a Stoicist background, okay? He's a Stoicist in his ethical views. I'm not going to go into what Stoics do, but basically they feel that there is a pattern of behavior that one follows that isn't directed towards hedonism. Put it that way. I think that's a good way to look at it for just, for the heuristic thumbnail understanding of what I mean by Stoic. He believed that God was the efficient cause, which means the divine agency in the world, that God was an efficient cause, actually made it happen. He's responsible, of course, not just for this metaphysics, but also for the Cartesian coordinate XY axis in all graphical formulation. Now, we didn't really see a good example of this in today's talk. Sorry. But every time you look at a graph where it's got the four quadrants, because you've got, an axis of X and Y, right? The ordinate abscissa, and you've got four quadrants. Normally, we only look at the one quadrant, right? The upper left, the upper right quadrant, right? But there are actually four. This is where you can do all kinds of hyperbolic analyses, and you can get all the information you want to get associated with sine and cosine, the slope of the curve, whether or not it hits the X origin or the Y origin. You can get all kinds of really important information by using his Cartesian coordinates. And he invented that. Yeah. He gave us also the rule of signs method for determining the number of positive or negative real roots of a polynomial. That's no small feat in mathematics. And he also invented, or at least he popularized, using superscript notation for showing the powers of exponents. That's all Rene Descartes. So finally he said, and I want to leave this with you, the senses can be in error. So what he did in his method, and then ultimately in his meditations, when he took his discourse and method, you should read this really fascinating reading, he stripped away all empiricisms initially to come to foundational truths. That's his discourse on method in one line. He is therefore the father of modern metaphysics, and he also led on the continental rationalism, which was basically met head on by imperialism, mostly of the British order. Of that we mean people like Locke, Berkeley, and Hume, which was all corrected back in the continent by Kant, of course. All right. So I also should mention that tomorrow's Easter, so happy Easter. Again, my name is Dan Guerra, and you heard another talk from VerEvMed Studios. Today we talked about the epigenetic patterning and Alzheimer's disease specific locus, the transcription factor, and the homeobox genes. Thanks for your attention. There's my Facebook page. There's my email if you want to contact me directly. There's the email for our company, and there's the website. Please go visit it, become our client. Remember, we are scientists verifying published evidence in the medical biosciences. All right, so we'll close this. We'll close off that entire thing there. We'll save it, of course, because we don't want it to go completely away. And I will give you my sign-off, of course.